Body odors can create immediate sexual attraction. Both men and women of various cultures seek out perfumes which highlight their body odor in the most desirable way. The famous novel Perfume by Patrick Susskind describes a perfume made of body odors that drives people into ecstasy and makes them forget civilized behavior. However, recent research indicates that the olfactory match between people rather than a universally irresistible smell might be the key to olfactory attraction. As early as the mid 1970s, mate choice in mice was shown to be influenced by genes at the MHC locus. This was followed by investigations in other rodents, fish, and birds, all with similar conclusions. Thanks to results from several studies conducted in the last 15 years, it looks like humans could be added to this list. However, questions remain as to the precise evolutionary pressures, the mechanism, and the magnitude of this effect, among others, in influencing mate choice in humans. In animals, immunologic compatibility determines individual variation of mate choice and is partly conveyed by olfactory cues. The individual genetic major histocompatibility complex MHC represents a part of the adaptive immune system. MHC molecules are located on the surface of cells and play a key role in discriminating between endogenous and exogenous potentially pathogenic material. Genes of the MHC gene cluster are expressed in a co-dominant way. When given a choice, mice prefer mates with dissimilar MHC molecules compared to their own. This is advantageous from an evolutionary point of view. The co-dominant expression increases the chances of high MHC variety in the offspring of MHC dissimilar compared to MHC similar couples, which in turn leads to enhanced resilience amongst a variety of pathogens. MHC-related mate choice is guided by the sense of smell in vertebrates. Parts of the heavy chains of the MHC glycoproteins are detected in various body fluids like saliva, urine, and sweat. In addition, it has been suggested that MHC molecules influence microorganisms of the skin which are involved in the formation of body odors. The precise mechanisms by which those MHC molecules shape the body odor are still under discussion. However, MHC determined odors activate vomeronasal and olfactory neurons. In terms of evolutionary pressure, local pathogens play a significant role in maintaining MHC diversity in the population and in selecting for specific alleles. As we now appreciate, this is because the MHC influences immune responsiveness. Due to its role in selecting and peptide fragments that will be presented, the inheritance of specific alleles at particular loci can predispose individuals to either enhanced susceptibility or resistance to specific infectious agents and immune disorders. Over long time periods, endemic local pathogens exert evolutionary pressures that drive higher or lower than expected rates of certain MHC alleles in a population, as well as overrepresentation of other resistance associated genes. The degree of diversity at the MHC locus clearly influences susceptibility to disease in populations. For instance, the European introduction of the smallpox virus to New World populations is credited with wiping out large Native American groups. This may be due to a lack of past evolutionary pressure for conservation of resistance-associated MHC alleles, which would therefore be rare or non-existent in this population, as well as to a lack of any individuals with immunity from prior infection. But How does an individual evaluate how well a potential mate will contribute new MHC alleles to one's offspring, leading to greater diversity and the potential for enhanced fitness? The primary candidate is odor. The MHC is known to influence odor in many vertebrate species. For example, the urine of mice from distinct MHC congeneric lines can be distinguished by both humans and rodents. Mice show a distinct preference for mating with animals that carry MHC alleles that are dissimilar to their own. In terms of maximizing the range of peptides that can be presented, this makes clear evolutionary sense, as increased diversity at the MHC should increase the number of different pathogenic peptides that can be seen by the immune system, increasing the likelihood of effective anti-pathogen responses. To back this up, the advantage of overall MHC diversity has been shown experimentally in mice, where most simulated epidemic experiments have found a survival advantage for H2 heterozygous animals over their homozygous counterparts. In humans, research in HIV infected individuals has shown that extended survival and a slower progression to AIDS are correlated with full heterozygosity at the HLA class 1 locus, as well as absence of certain AIDS associated HLA-B and C alleles. This specific link to class 1 is not surprising in light of the key role of CD8 T cells in combating viral infections. Human studies of attraction and mating also point to preferences for individuals with MHC dissimilar alleles. In one key study involving what is commonly known as the sweaty t-shirt test, college-age volunteers were asked to rate their preference or sexual attraction to the odor of t-shirts worn by individuals of the opposite sex. In general, both males and females preferred the odor of t-shirts worn by individuals with dissimilar HLA types. The one key exception was seen in women who are concurrently using an estrogen-based birth control pill. They instead showed a preference for the odors of MHC similar individuals, suggesting that this hormone not only interferes with this response but that it potentially shifts the outcome. But how, you may ask? Soluble forms of MHC molecules have been found in many bodily fluids, including urine, saliva, sweat, and plasma. However, these molecules are unlikely to be small or volatile enough to account for direct olfactory detection. Other hypotheses for how MHC might influence odor include via olfactory recognition of natural ligands or specific volatile peptides carried by MHC molecules, or by MHC-driven differences in natural flora, which are also known to impact body odor. The recent finding that some polymorphic olfactory receptor genes are closely linked to the MHC may also help explain this apparent association with mating preference. To date, the jury is still out on which mechanism or likely accounts for MHC-specific odor differences. As one might imagine, large methodological challenges are inherent in asking questions related to mate choice in humans, where differing levels of outbreeding as well as social and cultural factors influence the outcome. Nonetheless, it appears that, like most primate species tested so far, we humans may also have the capacity to use smell as an evolutionary strategy for promoting a robust immune response in our offspring.